So I'm Bluma Lesch. I'm an assistant professor in the genetics department, and we study chromatin. We mostly study chromatin in the germline, but we also look in other contexts. And what I just want to actually start off by talking about just the lab environment, what we do, and kind of what our priorities are. And I'm showing this is also my acknowledgement slide. Because people are very important to me. I just want to make the point that mentorship is important. I'm happy to sort of work with you to figure out what your ideal career is and get you there, work on presentation style, writing, and so on. And we have a really diverse group of interests and sort of priorities. In terms of environment in the lab, we're a genomics lab. We're also a heavily experimental lab. So basically every project in the lab generates genomics data. It's usually pretty interesting genomics data. Occasionally it's vanilla sort of RNA-seq stuff, but we have interesting intersections. And we focus on chromatin in the germline, which has two major biological implications. One is inheritance across sort of 1 to 2 to 3 generations, which I'll get into later. And the other is evolution, which I'll also get into in a few minutes. Our sort of overlying theme is to ask the right questions in the right way. We have a strong biological motivation, so we have a set of interesting questions. We have the ability to generate our own data to answer that. And then when we come to data analysis, we want to ask the right questions, not just sort of standard approaches to analysis of these data. So with that, I'm going to kind of zoom through about 4 short projects in the lab just to give you a flavor of the kinds of things that we're doing. The first 2 will be very short and the second 2 will be slightly longer. So this first one was actually published last year, and this was looking at a histone modification called K4 monomethyl. And this is a mark that's traditionally strongly associated with enhancers. It's actually often used to define enhancers. Sunny Bai noticed, not for the first time, that K4 monomethyl is also at promoters. And she also noticed that it has a very unusual distribution around promoters. It has this bimodal distribution, which you never see for any other histone modification, basically. And what she found, to make a longer story very short, is that if we split promoters based on their chromatin state, not transcriptional state, you can actually resolve this distribution into either bimodal or unimodal based on whether they're epigenetically poised or active. And this is actually not related to expression level, it's related to the chromatin state. We think this has implications for epigenetic inheritance. K4 trimethyl is very poorly inherited across mitosis. K4 monomethyl may be better inherited, and I'm happy to talk more about that if anyone's interested. A second short project, not a short project, but a short presentation of one, is also looking at sort of a non-canonical role for another histone modification, K79 trimethyl. This is a modification that's usually associated with active chromatin, but we actually found by IF that it's distributed in the nucleus in a way that looks like it's associated with repressed chromatin. So we went back to the genomics data, found and looked specifically at repetitive elements, and found that it's strongly enriched at pericentromeric repeats, generally missed because most people who are plugging through this data don't really look at repetitive elements. And then we found if we knock it out, we see a strong decrease in expression of these elements. This has major implications for control of the cell cycle, for early embryogenesis, and actually for predicting drug efficacy in cancer. There's a therapeutic drug that targets the methyltransferase for this mark, and we think we might be able to predict which patients will respond to it based on the response of expression of these elements. Okay, so the major theme of our lab is actually looking at chromatin across generations. And so I'll slightly expand that in the next couple of projects I'll talk about. And I just want to make the point that the majority of studies that look at chromatin look at this interval between the zygote, the fertilized egg, and the adult. This is colloquially known as development. But we're actually interested in the connections between chromatin and gene expression around this entire cycle. So what happens in the developing gametes, how does that affect the next generation at fertilization, how does it affect the adult, and how does that actually get transferred to the next generation. We're doing that by looking at paternal mutants for epigenetic regulators. And the reason we can do that is because males only have one X chromosome. So we knock out a gene in the male germline. We look at their sons. Their sons get their X chromosome from mom, and so they're genetically wild type. And so phenotypes in these offspring, we presume to be epigenetically inherited from the father. When we did this with a particular chromatin regulator, which is interesting for a variety of reasons that I won't talk about now, we see a significant difference in survival. This is in mouse. And that difference is associated with an increased tumor burden in these mice. So this is a huge black box between the time we make the initial perturbation and the time we see this phenotype in adult offspring. And we're trying to basically work through this black box now and figure out what's going on mechanistically. We see a major redistribution of a histone mark called K27 trimethyl. This is expected. We know this is an effect of the gene that we knocked out, although we can't connect it to the phenotype just yet. And we also see a major change in DNA methylation, which was more unexpected because there's no sort of known connection between the knocked out gene and DNA methylation. Most interestingly, we see that a lot of regions gain DNA methylation, both in the knockout sperm and in the bone marrow of the offspring, meaning it may be transmitted across generations. And these tend to be enhancers that are associated with tumorigenesis. Most interestingly, we can actually look at methylation in the sperm of offspring, and we see that some of these methylated sites, hypermethylated sites, are transmitted into the sperm, which may mean that they make it across multiple generations. And if we look at the distribution, actually we see a much higher spread in the sperm of offspring than we see in the knockouts. This is exactly what we'd expect, which is really cool, because basically a sperm is digital, right? A single sperm fertilizes an egg. That sperm is carrying one epigenetic state. If the initial spread is random in sperm, then we have to look at multiple offspring to see the effects of that initial distribution. And that's exactly what we're seeing. And here we see, just as a summary, we see about 500 sites where this hypermethylation is carried into the offspring sperm. This is an example at a tumor suppressor gene where we see a gain of methylation in the offspring sperm as well as the knockout sperm. So we're trying to figure out what sequence or functional features determine this retention of methylation, transcriptional consequences, both in the early embryo, adult tissues, and tumors, and the connection to the increased cancer susceptibility. All right, so in the last couple minutes, I'll talk about kind of the last implication, or the other major implication of intergenerational chromatin that we're interested in, which is what happens when we go around this reproductive cycle millions and millions of times. So how does chromatin regulation interact with evolutionary mechanisms, and how does chromatin state itself evolve? To start to answer this question, I collected a really large data set we published this about 4 years ago, of ChIP-seq data from these 5 mammals plus chicken as an outgroup. And I'm going to skip this slide just to say that germ cells are kind of an ideal cell type for this, because you can isolate precisely comparable cells from multiple species without needing reagents. And we're especially looking at this state called bivalent or poised chromatin, which is characterized by 2 histone modifications, K4 trimethyl and K27 trimethyl. This is a state that's strongly associated with pluripotency and developmental potential. So when we looked across species, we see that about 400 genes conserve this poised state. These tend to be developmental regulators that are central to conserve developmental networks. And so that's interesting. It implies that poising in the germline is related to control of somatic developmental networks. There's a connection between germ cells and somatic tissue. But it doesn't actually tell us much about evolution, just tells us that this is important. To look at evolution, we actually have to look at differences between species, and that's a lot of what the lab is doing now. I'm showing here two examples where a single species pops out as very different from the other species. And experimentally, we're following up on some of this to see what happens if we swap the promoter sequence from one species into another. Can we change the chromatin state? What are the sequence features associated with that? What's sufficient and what's necessary? We're also interested in cases like this. So I'm showing two examples where human, which is in blue here, has gained this poised state, this double positive state, K4 and K27. And rhesus appears to be in an intermediate state. So we can start to trace the evolutionary history of this state. In this case, we see a gain in the K27 mark before we see a gain in the K4 mark. And here there seems to be sort of a partial gain in both marks before we see a strong gain in both. So we're really interested in this question of what happens to gain or lose in a chromatin state across evolutionary time. There are actually ways to quantitate this. These have mostly been developed for quantitative traits, but have recently been applied to RNA-seq data. I think I only know of one reference where they've actually been applied to ChIP-seq data. So this is a really open area with a lot of room for development. So our major questions, what are the evolutionary intermediates for gain of a specific state? Does evolution of one mark affect evolution of another? And how does this relate to evolution of gene expression? We have all of these data, but we need to actually do the work to analyze it. And do dynamics differ at different types of genomic elements? And how do we quantitate this? So I'll end with that. And as sort of a final plug, I'd love to get rotation students. We have big biological questions. We think about gene regulation, which is obviously a really big and important topic. We have interesting new data sets. And if you're interested in an environment where you'd be working alongside wet lab scientists, that's what we are. So